Hello everyone. Welcome to Deeksha online classes. Today we are going to learn a new topic named animal diversity. Before we get into the topic, we will give a gist behind the classification. Now, animal kingdom is a part of 5 kingdom classification which was given by a scientist named R. H. Whitaker. According to R. H. Whitaker, he mainly considered nutrition as the main source of division, and he also took care of the type of nucleus, the morphological structure, the anatomical processes, and divided the entire population of entire organisms into Monera, Protista, Mycota, then came Plantae, and lastly came Animalia. Monera are bacteria. These are unicellular prokaryotes. Amongst the all other kingdoms, only Monera happens to be a prokaryote. Others are all eukaryotes. Protista is also unicellular, but Protista was formed separately from Monera because Protista was eukaryotic unicellular organisms. Plants are green photosynthetic forms, autotrophic in nature, whereas animals were heterotrophic. They could locomote. Mycota was a question before 5 kingdom classification came. This 5 kingdom classification main part which came out was of fungi. Fungi happens to be a saprophyte, meaning it grows on dead and decaying organism. It looks like a plant, but it is non-green, and as we can see, nutrition is also different. This is why the 5th kingdom Mycota came into existence. Now came another scientist by named Calvus. According to Calvus, he gave 3 domains of life. What are the 3 domains of life? What was it depending upon? It was depending on ribosomal RNA. In short, we called it as rRNA. There are 3 different types of organism based on ribosomal RNA. One had 16S rRNA, the other had 70S rRNA, and the last one had 80S rRNA. 16S rRNA was present in Archaebacteria. 70S rRNA was seen in case of Eubacteria, and 80S rRNA was seen in case of eukaryotes. These were the 3 domains of life given by Calvus. Now, if we have to compare this 3 domains along with our 5 kingdom classification, what do we understand? Eukaryotes comprises of Protista, Mycota, Plantae and Animalia. 70S ribosome is present in eubacteria, which is a part of Monera. 16S rRNA is a part of Archaebacteria, which is also a part of Monera. So, if we were to follow the three domains of life, then we would have 6 kingdoms and not 5. Based on this, we can look into a chart. So, we are seeing kingdoms of life as Archaebacteria, Eubacteria, Protista, Fungi, Plantae and Animalia. Now, let us get into detail of Animalia. Kingdom Animalia. Is divided mainly on basis of number of cells. Then we call it as protozoa and metazoa. Proto, the word signifies primitive or first. What kind of cells do you expect in the early stage? Prokaryotic. Zoa signifies animal. So, it is a primitive animal or the first formed animal or a prokaryotic animal. Surely the animal is unicellular. Then came metazoa. Metazoa was further divided into 2 criteria. One was eumetazoa, which means true multicellular animals, because meta means multicellular, zoa means animals. Besides the true metazoas, there existed few parazoa. What does para means? Beside. Beside the eumetazoa lies the few parazoas. Eumetazoa was divided based on symmetry. There are 2 types of symmetry. One in which when we cut a organism in 2 halves through the center of the plane, we get mirror image. We can cut in various positions, but the line should pass through the center. Such a symmetry is called as radial symmetry, and the classification is called as radiata. The other symmetry is a bilateral symmetry, where the cross section can happen only through one plane. We call such a symmetry as bilateral symmetry, and the classification is bilatria. Radial symmetry has 2 parts or 2 phylums. The first one is Coelenterata. The second one, or even Coelenterata, the other one name is Cnidaria. The other type of radial symmetry is Ctenophora. It is also called as Cnidaria. Earlier, the scientists considered Coelenterata and Ctenophora to be as a single phylum Coelenterata, but there are 3 very important features which distinguishes Ctenophora from Coelenterata, because of which it is separated into a separate family. From Bilateria, all the organisms are triploblastic. Until such time, the organisms were diploblastic. Now, these triploblastic organisms were divided based on coelom or body cavity. So, there are three different types based on this. The first one is acoelomata, or in other words, where coelom is absent. Next came pseudocoelom. Here in pseudocoelomata, coelom is present, but it is not a true coelom but a false coelom. Then came eucoelomata. So, we will learn one by one. Acoelomata, absence of coelom, makes the organism flat, and hence they are called as flatworm. This consists of a phylum called as Platyhelminthes. Pseudocoelom has a false coelom, making the organism roundworm. It consists of a phylum Aschelminthes. It is also otherwise known as nematodes. Eucoelomata, true coelom present, is now further classified into other phylums based on specialized characteristic features, out of which the first one is Annelida, second Arthropoda, third Mollusca, fourth Echinodermata, and fifth is Chordata. Annelida, the word comes from annulus, which means ring. If you look at Annelida, their body looks something like this. Each one is called as a metamer, and hence they follow metamerism. Arthropoda, on the other hand, has jointed legs. Poda means legs. This is otherwise called appendages. Next thing what we are going to learn is mollusca. These are shelled organism. The next phylum is Echinodermata. Echino means spine. Dermata means skin. So, spiny skinned organism. Till here, these all organisms are classified as invertebrates or non-chordates. The last one, chordata, completely depends on presence of notochord. Now, let us see in the chart until here. So, as we were discussing, Animalia is divided into Porifera, Cnidaria, Platyhelminthes, nematode, Annelida, mollusca, arthropoda and echinodermata. These are all invertebrates. Chordata, since it has notochord, comes as a very special phylum. Phylum Chordata is divided based on the locomotory organ. The first one is based on fins and second is based on tetrapod. Fins are present in fishes, Pisces, and Pisces are of two types, Chondrichthyes and Osteichthyes. Chondrichthyes has cartilage as its endoskeleton, whereas Osteichthyes have bone as its endoskeleton. Tetrapods on the other hand is divided mainly based on habitat. In case of tetrapods, the first one is amphibians. Second, reptiles. Third, aves, and last comes as mammals, in which we also are included. Amphibians live in both land and water. Reptiles prefer land. Aves prefer aerial mode of habitat. Mammals live in all air, or rather I would write it as arboreal, that is tree dwellers, land and water. Their most important characteristic is presence of mammary gland. So, you can see here Chondrichthyes, Osteichthyes, amphibians, reptiles, aves and mammals. So, in today's class we will be concentrating on three most important invertebrate phylums such as Porifera, Coelenterata and Ctenophora. So, in short, we will be completing till radial symmetry. So, let us start with Porifera. Porifera are pore bearing structures. They are sessile or sedentary, what we mean. So, if this is a Porifera structure, it is fixed to the ground. It is sedentary or also known as sessile. Porifera has small spicules throughout the body. This spicules is also called as spongial fiber. Throughout the body, Porifera has pores, and at the top it has a larger pore. These smaller pores are called as ostia. The larger pore is called as osculum. Porifera has a very important characteristic that it has only loose aggregation of cells. In other words, for the first time unicellular organisms aggregated to form a multicellular organism. In such a scenario, you can expect the cell lines in the dermal region. There are two cell lining. One is dermal and the other is gastral line. So inside this, so here also there is a 2 layered cells being present. This region, this entire region is called as spongocele. Spongocele at the top opens as osculum, and osculum is lined by a layer of cell called as choanocyte. Choanocyte is otherwise called as collar cells. Now this is in general the structure of a porifera. So let us see it step by step. As I have already told, they lead a marine habitat. They are sedentary, pore bearing structures commonly called as sponges. You can imagine that something has small pores which would look like a sponge, and hence the name sponges. I also have told you all the importance of loose cell aggregation. Choanocytes or collar cells line the spongocele, or which forms the water canal. Now what is water canal? Now again, looking back at the structure, this is our sponge which has a ostia through which the water is moving, and here is our osculum through which water comes out. When the water is moving in, it carries along with it dissolved gases and nutrition. When it is moving out, it carries excretory waste. So goes to say, sponges have water canal system that transport water, food and removal of waste. So I hope you have understood this first set of characteristics in sponges. We move on to the next. Water enters through ostia and into the spongocele and exits through the osculum. So body has numerous small pores called as ostia, which is a larger pore, osculum, singular and plural oscula. The entire body has a exoskeleton. What is exoskeleton? It is mainly for protection outside the body. Since we understand Porifera is sedentary, to save itself from all predators, it has spicules around. These spicules are nothing but spongin fibers. It is either silicon derivative or calcium derivative. Some of the examples are Sycon, Euspongia and Spongilla. So in this diagram you can easily see how the water is moving in through the ostia and moving out through the osculum. Continuing, we understand reproduction. These organisms, I want to repeat once more that they are sedentary, meaning they cannot move for whatever reason. So obviously they cannot find a mate. So they are hermaphrodites. What do you mean by hermaphrodites? Bisexual organism. Sexes are not separate. They are present in the same organism. This organism, we need to understand, has just now formed into a multicellular organism. So hermaphrodite is a good choice for them. Since they are hermaphrodites, fertilization can be internal. Male gamete also has been formed inside. Female gamete is also formed inside. So fusion occurred inside the organism and formed into zygote. But this zygote did not grow into an adult. It formed into a larva, then into an adult. Since the process is indirect, we call it as indirect development. If there was no larva present, then I would have named it as direct development. There could be another form of reproduction, that is asexual. We need to understand that these organisms have just now formed from unicellular organisms, and the main mode of reproduction in unicellular organisms are basically budding, fragmentation, and spore formations. So, in case of Porifera also we see these. So, in the first example, we see a small bud attached to a bigger Porifera. This bud detaches and grow into a new Porifera. In second part, some cells of Porifera shed and grow into a complete new Porifera. This is called as fragmentation. And in spores, we have specialized spores called as gemmules, which is grown internally in unfavorable condition of the organisms. These spores come out and grow into a new individual. So, can I say that budding is exogenous, whereas gemmules is endogenous? These are the three methods of asexual reproduction also seen in case of Porifera. Moving on, we see a few examples: Euspongia, Cycon and Spongilla. Now, we are moving on to the next phylum, phylum Coelenterata. Phylum Coelenterata, also known as Cnidaria, is a more evolved form of phylum. This phylum has several characteristics which is very important for any animal. For example, that this phylum, some of them are sessile just like Porifera, but some others are very much motile. So, motility is a very important characteristic for animals. These are also aquatic. Very important, they are radially symmetrical, diploblastic animals. They have various cell organizations. But before I move into cell organization, I would like to make you understand how they look. Now, as we have strictly told that some are sessile, some are free living, we understand them properly. The ones which is sessile are called as polyp. They look similar to Porifera but with tentacles around them. But on the other hand, medusa, which is a free swimming one, looks like a Portuguese hat, and Portuguese hat is known as medusoid. A Portuguese hat along with the head hair looks something like this, and in this case our free swimming medusa also looks like this. The tentacles on them had some specialized cells which were called as stinging cells. These stinging cells, otherwise called as cnidoblast or cnidocyte, is the main reason why we call phylum Coelenterata to be as Cnidaria. Now, these cnidoblasts or the stinging cell help in anchorage, catching of prey and escaping predators. It is very important for us to know that these organisms help in catching the prey by itself, not like Porifera who depended on water. So, this is very important. Now, when you are seeing these cells moving inside, there is a cavity which is formed. This cavity is called as coelentron and hence the name coelenterata. This cavity is not the body cavity. Let me be very clear. Here we are diploblastic animals. So, there is no body cavity. This forms a gastrovascular cavity with a single opening near the mouth region. So, I hope this slide is very clear to you all. Moving on, we see the polyp and the medusa structures. Now, certain coelentrates are specialized. The specialization is deposition of calcium carbonate as exoskeleton. Again, if you can really relate it, Porifera also had spicules which were calcium or silicon derivative. These are corals, a very important part of coelenterata. Now, the most significant feature what we see in coelenterata is metagenesis. Metagenesis means alternation of generation. What are the two alternate generation? One is polyp, the other is medusa. You can see the polyp form is otherwise called as the coral form. Medusoid form is otherwise called as the jellyfish. Medusoid forms give rise to polyp. Polyp forms give rise to medusa. They alternate each other in their growth. Now, some cnidarians exhibit both the forms. One such cnidarian is Obelia, which has 100% metagenesis. But certain forms have certain amount of asexual reproduction also. The only mode is budding. Now, you can see here also there is an improvement. In the earlier case, we saw fragmentation, budding and spore formation as asexual reproduction. We all would agree that sexual mode of reproduction is a better mode of reproduction, evolved mode of reproduction. So, here we see only one form of asexual reproduction and we have given up all other forms. The most important form of reproduction stands as sexual reproduction. So, here is a structure or a life cycle of a Obelia. You can see medusoid Obelias give rise to sperms and eggs which fuses to form zygote. Through an indirect development due to the larval stage planula forms into a polyp Obelia. Some of the examples seen in case of coelentrates are Physalia, which is known as Portuguese man of war, Penatula, which is otherwise called as sea pen, Gorgonia, which is called as sea fan, Meandrina, it is called as brain coral. There is another phylum which is very similar to that of Coelenterata. For a very long time, scientists put Ctenophora under Coelenterata itself. The phylum is Ctenophora, which is otherwise called as Cnidaria. There are three distinguishing factors of Ctenophora from Coelenterata. Other than these three, all the points and characteristics are same. The first point is the name Cnidaria itself. The word Cnidaria in Coelenterata was given by Cnidoblast. In Ctenophora, Cnidoblast is absent. Instead, there is presence of Coloblast. Coloblast helps in anchorage, but not for stinging. These have comb-shaped structural forms of legs which is appearing in pairs of 4s. These are tentacles which appear like combs. Hence we call them comb jellies or sea walnut. These also, like Coelenterata, have radial symmetry. They are also diploblastic organisms and they have comb-shaped tentacles for their locomotion. So as I told, there are 3 points which differentiates. As of now, we have learned 2 points. One is presence of coloblast and absence of cnidoblast, and the other formation of comb jelly, comb plates for locomotion. Digestion is both external and internal. So absorption of food happens from outside, from water to the organism, and digestion further inside in the cellular level. The third point which I said is different from Coelenterata is bioluminescence. These organisms have a property where they can glow in water. They exhibit bioluminescence. Pleurobrachia and Ctenophora, Ctenoplana are 2 organisms which exhibits bioluminescence. So bioluminescence is exhibited by Pleurobrachia and Ctenoplana. Like Coelenterata, they also exhibit metagenesis, and hence you can see here medusoid forms giving rise to sperm and eggs forming zygote. A larva is seen, planula larva, which gives rise to polyp, and then back again into young medusa. So, I hope you have understood today's lecture. So, we are going to re-summarize whatever we have learnt. We have learnt different types of kingdoms. Then we have learnt classification of Animalia. Then we moved on to Chordata classification. Then we have also seen each invertebrate specially. They are Porifera, Coelenterata and Ctenophora. Hope you liked today's class, but after liking it, it also is mandatory that we start doing our homework. It reinforces all our concepts once again. So, while I do today's homework questions, I would like to clarify certain doubts of yours so that your homework becomes much easier. Now, name the phylum that represents acellular animals. Animal which is acellular, absence of cell. I will only be giving you hint. If you still have doubts while solving homework, in the online sessions I can clear it. Cytoplasm of protozoan is differentiated into. Now in this question, understand very clearly that a protozoan is unicellular. So one single cell is bound to do all the work. So here is our incipient nucleus, here is our cytoplasm, which exhibits cytosolic movement for circulation. So you can expect what are the two types of cytoplasm, one which is outside ecto, one which is inside endo. So based on that we have options are A, endoplasm and epiplasm, B, ectoplasm and mesoglia, C, ectoderm and endoderm, D, ectoplasm and endoplasm. Third question, choose the locomotory organ in Plasmodium. Here I would like to explain the classification of protozoa. Protozoa has 4 classes based on locomotory structures. First one is rhizopoda. What do they have? They have pseudopodia. Next, flagellata. I think the name itself signifies presence of flagella. Then you have ciliata, cilia present. Then you have sporozoan. These do not have any locomotory organ. Now you have to find out what is Plasmodium, whether it is a rhizopoda, flagellata, ciliata or sporozoan. Next comes osmoregulation. What is osmoregulation? It is balancing the water content in the body. So how you are balancing? Along with electrolytes. Now let me explain you with an organism. This is Euglena. Euglena has one single flagella. It is generally greenish in color, covered by a layer which is called as pellicle. At the base of this flagella, there is presence of contractile vacuole which helps in maintaining the water balance. So based on that question, we see here what is osmoregulation. It is a process of maintenance of water balance. B, it is a process of maintenance of salt balance. C, it is the process of maintenance of water and salt balance. D, none of the above. Next question, which one of the following is osmoregulatory in protozoans? So I think the previous question it is explained. So I am not explaining this question. Try to answer. A, contractile vacuole, B, cell membrane, C, food vacuole, D, cytophage. Question 6. Organisms which have more than 2 types of nutrition are called A, autotrophic, B, heterotrophic, C, mixotrophic, D, saprozoic. So I was speaking of our Euglena. It has contractile vacuole, it has cytoplasm, it also has a pellicle covering. I also told it is sometimes green in color. So green signifies that the moment it is getting sunlight, it shows photosynthesis. But this is a very lowly organism. If it is not getting sunlight, it shows chemotropic nature. So what happens? Together it is called as mixotropic. So, based on that, try answering this question. Organism which has more than 2 types of nutrition are called as autotrophic, heterotrophic, mixotrophic or saprozoic. Question 7. Which mode of nutrition is applicable to protozoa? Holophytic, parasitic and saprozoic, holophytic, parasitic and saprozoic. Now, this depends on how many organisms do you know. Let us try to explain some of them. Now, there are organisms which are green in color. That will be obviously autotrophic. We know certain bacteria cause ill health, parasitic. Certain organisms or unicellular forms live in aquatic conditions, which is called as zooplanktons. These zooplanktons consume another zooplankton or a phytoplankton, when we call it as holozoic or holophytic. Based on this, you can try and answer the question. Next question, mixotropic nutrition is seen in Euglena, amoeba, opalina and plasmodium. The question hint is given in previous questions. Try answering it. Next question, how does respiration and excretion takes place in protozoans? First option, general surface of body, through contractile vacuole, through oral grooves or through cytophage. I want to again highlight the fact that protozoans are unicellular. So, based on this, think of the organism, how would they respire and excrete. Asexual reproduction in protozoans takes place by binary fission, budding, multiple fission, all of these. In all protozoans, cell divides by asexual method only. So, this is the hint, try solving it. 11th question, which one of the following is mode of sexual reproduction in protozoans? A, fusion of gametes, B, conjugation, C, both of these, D, none of these. Now, I will only ask a question instead of in case of a hint. Can you identify a male bacteria and a female bacteria separately? If you can, your answer would be fusion of gametes. If you cannot, your answer will be conjugation. So, moving on to the next question. Question number 12. The name the Porifera is justified because of the presence of. So, why do we call a Porifera as Porifera? Because of canal system, because of ostia, because of choanocyte or spongin fibers. Question number 13. Large opening for the exit of water in poriferans is called as spongocele, dermal pores, ostia or osculum. Question number 14. Endoderm of coelenterata encloses a digestive cavity called alimentary canal, gastrovascular cavity, pseudocele or gastrocele. Last question. Stinging cells in cnidarians are A, choanocyte, B, monocyte, C, nematoblast and D, A and B both. All the best students. Do your test well. We will get back in online session.